So in the second part of Chapter 8, we're going to focus on what they call bioenergetics, which sounds like a really fancy word, but basically it's just the study of cellular energy release. And so what you're seeing on the screen here, and this actually begins on page 236 of your book. 237 has a really good image. It's figure 8.16 that I'll be pointing out a lot, and you probably want to have in front of you to kind of reference. But so what you'll see on the screen is the major pathways that we see as far as what most cells utilize in order to generate energy. And so for most cells, typically going to begin with glycolysis, which you'll recall from biology is the breakdown of the sugar glucose. And so glucose being the major fuel source for energy, it's not the only energy source, fuel source that can be used for energy, but it's the main one, so that's typically the one that textbooks are going to highlight. So it begins with glycolysis. From there, most cells move into the Krebs cycle. Some textbooks also call this the citric acid cycle. We'll refer to it as the Krebs cycle in this class. And then finally, it's carried out with what we call the respiratory chain. In biology, we call this the electron transport chain. So it's the same thing, just a different terminology that they use in here. Now, in order to do those pathways, the glycolysis, the Krebs cycle, the respiratory chain, or the electron transport chain, there are actually three types of options as to how cells can kind of go through this. And so if we'll look at these catabolic pathways, just kind of giving a quick difference, and then we'll look into some more detail behind these. So the first option that microbes have is the same option that we have, which is aerobic respiration. And so if you look at that word aerobic, that means that it's going to require oxygen. And so for aerobic respiration, you'll notice it's those three pathways that we just listed. It's glycolysis, then the Krebs cycle, then the respiratory or electron transport chain. The benefit of aerobic respiration is it's going to generate the largest number of energy every single time. Now, our biology book and our microbiology book give different numbers of ATP. In biology, it references 26, but in our micro textbook, it says 38 possible ATP. So for this class, let's just learn the number 38. But again, what that means is a large amount of energy per glucose. Now, one thing that is required for aerobic respiration is that it does require oxygen to be the final electron acceptor. We'll talk more about that in just a second. So there is a second option for microbes, which also is a second option for eukaryotes as well as prokaryotes, is anaerobic respiration. So again, you'll notice it follows that same pathway of glycolysis, Krebs cycle, respiratory chain. It's still going to generate energy, but not necessarily as much as aerobic. You'll see that it says that it can vary in how much ATP is being generated depending on the microbe that we're talking about. Now, because it's called anaerobic, if you put an in front of a word, it means without. So anaerobic is without oxygen. It does not utilize oxygen as its final electron acceptor. It's going to utilize some other type of source there. And then finally, a third option that microbes have, again a third option that eukaryotes and prokaryotes have in common, is fermentation. And so we know fermentation actually only utilizes the first step in most pathways. It just uses glycolysis, it breaks down the sugar glucose, and uses it to generate a small amount of energy. Kind of the downfall of fermentation is it's only able to produce 2 molecules of ATP, which again, not typically enough to sustain life, but it does give you a rapid burst of energy when there is no oxygen available, and so you're just trying to really quickly generate ATP. So again, if you look in your book, you'll see this on page 237, and again, just highlighting what's going on in these three options of catabolic pathways. So we have aerobic, where you got glycolysis, you have Krebs cycle, you have electron transport, reminding you that oxygen has to be available, and it's going to generate 38 molecules of ATP. There's anaerobic, again with glycolysis, with the Krebs cycle, with electron transport. Biggest difference you see here is that it's not oxygen to be the electron acceptor, it's some other type of molecule, and then again ATP numbers can vary. And then again, fermentation, only relying on glycolysis to generate small amounts of energy. So one thing you'll want to make sure that you note is just what makes these three pathways different, also noting what they have in common. They all three begin with glycolysis, and then when you look at the first two types, aerobic and anaerobic, they're basically the same process except for what's actually found at the end, whether it's oxygen or some type of non-oxygen electron acceptor. So if we take a closer look at these pathways, let's spend some time with glycolysis first off. Now, if you recall, glycolysis again is going to take the sugar glucose and it's going to break it down. Now, remember, glucose is a 6-carbon molecule. And so if you take anything that there's 6 of and you split it in half, you're going to generate 2 groups of 3, right? So if we have a 6-carbon glucose, we split it in half, it gets oxidized, meaning it's going to lose electrons. In that losing of electrons, it's going to split into 2 groups of 3. We call those pyruvic acid. Okay, so at the end of glycolysis, we've taken glucose, split it in half. We call those 2 groups pyruvic acids. And since electrons were given off, it's going to produce something called NADH. Now, if you remember from biology, NAD and FAD were great examples of taxicabs, right? They are electron acceptors. So when you split a molecule and electrons are released, NAD and FAD are going to catch those electrons, which means they're going to catch the hydrogen that they're part of. And so we write it now as NADH because it's carrying those electrons. Okay. It also mentions the Krebs cycle here. And then quickly, electron transport. So I did just want to remind you, just to kind of see what's going to happen here, right? So we got glucose, it's being split into the 2 pyruvates. We're going to generate a small amount of energy here because we are splitting sugar, and again, we're going to make NADH. But again, this is only step 1 in aerobic respiration. We then have to follow up with the Krebs cycle. Okay, so these pyruvates are actually what link glycolysis to the Krebs cycle, because we're going to take those pyruvates and we're going to want to use those to continue this process, right? So we're going to take those pyruvic acids and we're going to convert them into something called acetyl-CoA. So it's acetyl coenzyme A. What happens here in this process is that the 3 carbon molecules get decarboxylated. Sounds real swanky, but basically just means they lose a carbon. That's converted into carbon dioxide, which is typically expelled as a waste gas, right? But in the process of doing this, it's going to generate a small amount of energy. So it's going to generate 2 more molecules of ATP because, again, we took the 6-carbon glucose, we split it to 2 groups of 3, right? So when we convert those into acetyl-CoA, again, we still have 2 groups, so this process is going to have to turn twice. So if one group generates an ATP and the other group generates an ATP, 1 plus 1 equals 2. Okay, so at this point we have generated a small amount of energy, but we want to generate more here, right? So the last thing we move into, and by the way, it does mention that this is a part of oxidative phosphorylation, right? Because it's an oxidation, or it's a redox reaction that's actually giving off the energy here. Okay, so then we move into the final step, and you'll notice that these objectives are kind of all, they're separated out, but they're kind of all smushed together. So in class, we'll make sure that we kind of clear up what you want to have for each of these. But then it also falls into the final step, which is the respiratory train, or you can call it the electron transport chain, where we take all of the NADHs and all of the FADH2s. These are all those taxicabs that this whole time have been collecting released electrons. And so this is the chance to process those electrons, to process the hydrogen and generate ATP. And so the way that it does that produces up to 34 molecules of ATP. So a lot of energy that's actually produced here. And so it mentions the way that it's produced here is it involves an enzyme, ATP synthase, which is again protein by structure, to kind of shuttle electrons down the chain. And as it moves electrons down the chain, energy gets released, that energy gets captured, and it's used to produce ATP. Now, this process that we'll talk about here that's used to generate the energy is actually called chemiosmosis, something that we used to talk about in biology. And so by definition, chemiosmosis, it's basically where the electron transport chain is going to shuttle electrons, As electrons move down the chain, hydrogens get pumped across a membrane. And anytime you take hydrogen, which has a positive charge, and you send it from one side of a membrane to the other, it's going to produce a gradient, meaning both sides of this membrane are no longer going to have the same charge. You're going to have more positives on one side than you do on the other. And as you produce that gradient, or that difference in charge, it's going to cause the formation of ATP. 34 molecules of ATP to be exact. Okay. Now, one of the things that we do want to point out here is that for eukaryotes, this process of chemiosmosis, this process of electron transport, is going to happen in the mitochondria, right? Because in eukaryotes, remember, mitochondria is the powerhouse of the cell, so it's the one that actually contains this electron transport chain, and it's going to produce those high volumes of energy. But now remember, in prokaryotes, they don't have a nucleus, and then they also don't have membrane-bound organelles. So you're not going to find a mitochondria in them. So the question is, where does this process of energy reactions actually occur? Well, it's actually going to occur in what we call the periplasmic space, or the intermembrane space. So if I show you a picture of this, and you can see this in your book, depending on what page you're at this point, but you can kind of see this when you scoot past the mitochondria on page 245. They'll show you within a prokaryote. See, here's your cell wall, right? That's the outer layer. Here's that cell membrane, and so this is the periplasmic space. So this is ATP synthase, this is that protein, and so as electrons get passed down the chain, as they get passed from one protein to the next, hydrogen is going to get pumped out. And so if you take this positive hydrogen and you move it from this side to the other side, you're going to end up with more positive stuff here than you do here. That's called an electrical gradient. And an electrical gradient is typically the on switch to generate ATP. So you start doing all this phosphorylation and you make large volumes of ATP. And then, of course, remember, oxygen required to do all of this, and so as oxygen is hanging out at the end of this chain, it's going to catch electrons, it's going to catch hydrogens, and when you combine oxygen with hydrogen, you're going to form water, which is the very last thing that's generated here. Now, to kind of sum all this up, just a quick reminder, so glycolysis by itself is going to generate 2 molecules of ATP. Remember, that's where we split the glucose, made the pyruvic acids, and we're catching electrons here, so it's going to generate 2 ATP. The Krebs cycle itself also going to generate a small amount of ATP, 2 of them, and some carbon dioxide, some more taxicabs that are filled. And then finally, electron transport going to generate that highest volume of ATP with 34. So if you take 2 plus 2 is 4, add 34, that gives you that grand total of 38 molecules of ATP.